Good afternoon, everyone. Thank you very much for your introduction, and thank you for the insight that you gave me over lunch. I am Anna Tagliaferri, and I am currently conducting my research at the Sensing Technologies Lab at the Free University of Bolzano. Today, I will present to you the work that me and my colleagues conducted on carbon nanotube electrolyte-gated field effect transistors, trying to improve their stability through the use of a lipophilic membrane. So just a bit of introduction on who we are. We are located in a very small town located in the northernmost region of Italy, that is Bolzano. It is located in the middle of the Alps and more precisely in the landscape of the beautiful Dolomites. This leaves us with a very good landscape and a very good outlet to go to when we're done working for the day, for the weekend. We can go on beautiful hikes and climbs. And I encourage you to visit at least once because it is very, very beautiful there. So in our laboratory, we work a lot on biosensors with applications in many fields, ranging from food safety and food quality, and where we have developed biosensor to detect foodborne hazards such as spermidine and histamine so far. Then we also have some biosensors for sport monitoring, where we developed a biosensor to detect ammonium ion in sweat, which could improve the athlete's performance. For example, I will show you a little bit later. And then we also have some applications in, which is here, the computer, it's not there, sorry, in health monitoring, where our biosensors could be integrated, for example, in wearable devices. And this could help us to early diagnose some inflammation cycles and processes on the skin in a non-invasive way. And finally, we also have some example of sensors used in environmental monitoring, such as, for example, in the case of nitrate residues coming from pesticides in free waters, such as the rivers around our town, but also for microplastics. So I will present to you a little subset of the biosensors that we have so that you can follow along our thought process in trying to develop this new project and trying to bring this to term. So first, I will give a brief introduction on electrolyte-gated carbon nanotube field effect transistors, explaining you our thought process in why we added this lipophilic membrane on the carbon nanotubes. I will show you how we fabricate these platforms, these devices. I will show how we electrically characterized the biosensors, trying to study their stability over time, how we validated their performance by the addition of an ammonium selective membrane. And at the end, I will summarize our results and I will give a little bit of insight into what can still be achieved with our biosensors. So to start, just a brief recap. Electrolyte-gated carbon nanotube field effect transistors are composed of three electrodes. First, we have the gate, which is the modulator of current, the current that flows between the source and the drain electrodes. Between the source and the drain, the channel is found. Our channel is composed of semiconducting carbon nanotubes, which are the main focus of the topic of the talk of today. Of course, for these devices to be functional, they need a dielectric material. In our case, the dielectric role is taken up by the electrolyte. The electrolyte of our choice is phosphate buffer saline, or PBS in short. Some of you might be wondering, how does this work with an electrolyte? So when no voltage is applied, the charges are randomly distributed across the electrolyte and no dielectric properties are present. But when we applied a voltage to the gate, the charges accumulate at the semiconductor and electrolyte interface and at the gate and electrolyte interface. This leads us to a capacitor behavior, effectively establishing the gate dielectric of the device. When we apply a further voltage to the drain, the charge carriers that have accumulated at the channel electrolyte interface start flowing, effectively giving us a current that can be measured as the device response. As I was saying, we have carbon nanotubes on our channel. So I'm sure that we all know this, but just as a reminder, carbon nanotubes are one-dimensional nanomaterials completely made of carbon that can be visualized as hollow cylinders. Among their excellent properties, they have a large surface area that can be exploited for the functionalization, as I will show you in one of our examples. The large area allows for a large number of biorecognition elements to be attached to them, helping us in improving the sensitivity of the devices. They also exhibit high carrier mobility, which makes them excellent semiconductor materials for our applications and our purposes. Moreover, they are processable, easily processable in solution with the addition of a surfactant, such as carboxymethyl cellulose or sodium dodecyl sulfate. And this here is an example of one of our water-based carbon nanotubes inks that we used for the spray coating deposition, that I will also explain later. On the other hand, they exhibit hydrophobicity. This limits their functionalization in a few instances, and that's why we moved away from the functionalization of carbon nanotubes, preferring the functionalization of the gate electrode. Since we are using a random network of carbon nanotubes, we also see a very large device-to-device variability, which has been somewhat mitigated by the deposition of the membrane. The main thing that we have tried to tackle here is the instability that they show. And by instability, I will show you the results that we obtained without the addition of the membrane. So as I was saying, I would like to bring you along our thought process very briefly, and I will show you some of the examples that we have developed already in our laboratory. One of the EG-CNTFET that we have fabricated is for microplastic detection. In particular, in seawater, we find a great number of polystyrene nano and microparticles. And for this, we have created an electrolyte-gated field effect transistor with a planar configuration, which we fabricated on glass substrate. And we functionalized the gate with a peptide that was found in literature to be selective for polystyrene nanoparticles. The work is in the process of being published. I do not want to spoil my colleague's work, but you can see from her results that we obtained a linear detection up to a concentration of 1 milligram per milliliter. I am hopeful that, and I'm sure that we will see her results being published really, really soon because her work was outstanding. Another device that we have fabricated was for spermidine. Spermidine is a molecule that is typically found in food and particularly could be dangerous in meat. Why? Because when in meat, it can react with nitrites, producing the dangerous nitrosamines, which are known carcinogenic molecules. In this case, the functionalization was different. This was the example that I was telling you before. We have conjugated the antibodies, anti-spermidine antibodies, with the carbon nanotubes by means of pi-pi interactions. You can see that for this example, we obtained a linear detection with a very high coefficient of determination, close to 100%, with a very wide linear range of detection from 0.001 to 100 nanomolar, and a limit of detection in the picomolar range. This was one of the cases where the carbon nanotubes were very unstable, and I will show you later what I mean by that. Further, step that we took was to drop cast an ammonium ion selective membrane on the carbon nanotubes. This led to a slight improvement in the stability of the carbon nanotubes, and we found a linear detection range, which is much higher, but it still covers the whole physiological range of ammonium that is produced in sweat during activity. The coefficient of determination in this case was also not great, but further improvements have been made, and a new device is being developed right now. What is important to note here is that this type of biosensor was tested in artificial sweat, and it still got a very high selectivity against the other molecules, the other ions that are present typically in sweat. Just recapping a little bit the problems that we obtained with these three devices. In the first two cases, we had the carbon nanotubes not protected by any kind of membrane, and what we experienced is that the current decreases rapidly over time. This leads to devices to stop working, which is less than ideal. Moreover, they are single-use, and we have to throw them away very quickly. For the third example that I have shown you, it takes 1 hour for the device to stabilize and to reach a state where the current is linear over time. After this, we have to leave the devices to equilibrate for another 40 minutes, which means that it takes a very long time to perform the measurements. And it is not feasible nor sustainable for our application of continuous sweat measurements on athletes. We cannot leave them cycling or running for 2 hours and a half before starting the measurements. And so it's from these premises that my work began. First, I started with the fabrication using standard negative photolithography. I chose a polyimide substrate, a flexible substrate. I deposited the photoresist by means of spin coating, exposed it to UV light through a patterned mask. I then deposited an adhesive layer of titanium using electron beam, followed by the thermal evaporation of gold. After 1 hour liftoff in acetone, these are the devices that I ended up with. Here, magnified, you can see just the detail of our interdigitated electrodes, where we sprayed the carbon nanotubes. This was our setup. The process was previously optimized since we had already several examples of carbon nanotube electrolyte field effect transistors. And we set the hot plate to 70 degrees Celsius. We set the distance between the substrate and the nozzle fixed at 5 centimeters, and the speed at 150 millimeters per second. After the spray, we had to check the quality. The first way and immediate way that we had was to check at the optical microscope. Of course, we cannot see the carbon nanotubes at this point. What we can see is the marks left by the surfactant. The size and the distribution of the marks left by the surfactant can give us a preliminary idea of the quality of the spray. To effectively check how the spray worked, we had to conduct, of course, other experiments and other analyses, such as atomic force microscopy. From the picture here, we can see that we have, of course, a random network of carbon nanotubes that are evenly distributed. There are no bundles, and therefore ensuring proper conduction and proper function of our devices. At this point, we had to go on with the electrical characterization. We did this by using a semiconductor device parameter analyzer, or probe station. And we performed the test using 200 microliters of 1x PBS. We had a mixture of salts and minerals dissolved in the solution. We measured 40 consecutive transfer curves or IV curves with no waiting time in between each other. This means that the transfer curves were collected over the span of 1 hour. We swept the gate voltage from 0.2 volts to minus 0.8 volts, keeping the drain voltage fixed at minus 0.1 volt. The results are seen in these plots here. So this is the result of one of our devices, the transfer curves that we have collected. And what we can see from here is that over time, after each cycle, the current decreases. To better see this phenomenon, we plotted the on current versus time. That is the highest current that we measured at minus 0.8 volt. This is seen in this plot here on the right. We can see that over time, the current decreases very rapidly in the phenomenon that I was talking about before. We only left our devices running for 1 hour, but it is plausible that if we had left them for longer, the current would have reached zero very quickly after this. We then tried to perform a linear fitting on this plot here to see how long it would take to our devices to reach a linear current over time. What we considered good linearity is when the R squared was higher than 0.98. So on average, we obtained a linearization time of 33 minutes. Of course, it is not enough to just test the time. We also had to verify how the performance changes over time. And we calculated the on-off ratio, the ratio between the highest and the lowest current that we have measured. For this, the current decreases over the span of 1 hour from 89 to 61, with an average worsening of the performance of 31%. After checking that the performance is worse, we then deposited our membrane, our insulating membrane, on the carbon nanotubes. The membrane is polyvinyl chloride based, and the lipophilicity is given by this ETH 500 compound. The membrane is there to insulate the semiconducting channel and to protect the carbon nanotubes from the environment, that is from the air and from the electrolyte. We had to play around a little bit with the volume of the drop casted membrane, and we found that the optimal way to actually cover the channel was to first drop cast 8 microliters of the membrane, let it dry for 30 minutes, and follow this by another drop casting of 7 microliters. And that is why you see the 2 markings here on the corner of the channel. Of course, we also evaluated the thickness by means of profilometry. We found that the average thickness of the membrane was around 60 micrometers, with some peaks at 80 micrometers. This technique suffers the coffee ring effect, which we are hoping to mitigate in the future by not drop casting, but blade coating this membrane on the devices. After the successful deposition of the membrane, we still performed the electrical characterization, collected the 40 transfer curves over time, and we saw that we obtained an opposite effect with respect to what we have seen before. In this case, the current increases over time. Again, to better see this phenomenon, we plotted the on current over time, and we see a very different plot with respect to what we have seen before. We see that first, we have a stabilization phase where the devices are still finding their equilibrium, after which we find the slope phase where the current increases. If you remember, before we had very rapidly decreasing current. Right now we obtained the opposite effect. If we plot the slope phase alone, we can see that the R squared is really, really high, higher than 0.99, and the current slowly but surely increases. We performed then the same parameters extraction as before, and we found that it takes on average 34 minutes for our devices to reach a linear current trend over time. And the performance also improves over time. We start from an on-off ratio of around 10, and after 1 hour, it more than doubles to a value of 25. So the overall improvement in the performance is close to 151%. Just to give a little recap of our results, we cannot really say that the devices with the membrane reach a stabilization point. They reach a linear trend over time, but not really stabilization. In our case, we reach this stabilization in 34 minutes, which is approximately half the time that state-of-the-art devices fabricated with polymer-coated carbon nanotubes exhibit. Also, the improvement in performance is really outstanding. We can see a decrease in the performance without the membrane encapsulation, and we see a 151% improvement in our devices. Of course, we had to validate this with some measurement of some analyte. We chose as proof of concept to measure ammonium. And first, we drop casted an ion-selective membrane on our gate containing an ionophore produced by actinomycetes, that is non-actin. After this, we had to leave the device's conditioning in 1 millimolar ammonium, since we had previously seen that this conditioning step was greatly improving the sensitivity of the sensor. We then measured 5 different concentrations of ammonium, and the response of the device was taken at a fixed gate voltage of -0.8 volts and a drain fixed voltage of -0.1 volts. We did 5 consecutive additions of ammonium every 10 minutes after the stabilization time, and we see that we get a linear response over the whole range of concentrations that we have tested, with an R-squared, with a coefficient of determination close to 95%. To conclude very quickly, the membrane increases the stability and the performance of our devices, as indicated by the improvement in the on-off ratio. Also, the membrane leads to the stabilization, effective stabilization of our devices in 34 minutes, improving much with respect to the state-of-the-art devices. And most importantly, the membrane does not interfere with the analyte sensing. We have successfully reached a linear detection of ammonium in the whole tested range. Where does this leave us? We have much work to do. We still have to improve the sensitivity of the biosensor. Of course, this was a proof of concept testing that the device would still work as a biosensor. In the future, we are also hoping to study the mathematical model of the devices, and after this to run simulations so that we could better understand the underlying working mechanism, and so that we could further optimize the process and possibly reach even better performances. I would like to take 30 seconds to thank all of my colleagues, all my group, as they have been of great help in running this project. And I would also like to steal a couple of your seconds to advertise that we have 2 available PhD positions for next year. And if you would like some more information, feel free to scan this QR code or to reach to us, write us an email, talk to me at the end of the talk. I would like to thank you very much for your attention. And if you have any questions, I will be happy to answer.